Hey guys, it's Kirsten. Today we're going to be talking about cell viability assays, specifically the MTS assay. What are cell viability assays? Cell viability assays are colorimetric methods for quantifying viable cells in a culture. Cell viability really just means living cells. So there's many different kinds of these assays, but we're going to be focusing on the MTS assay today. So what are these cell viability assays used for? They're useful for lots of different things. They can provide information on things like cellular proliferation in response to things like cytokines or nutrients or growth factors or mitogens. It can also give us some information about cytotoxic drugs and chemicals and other pharmaceuticals. It will give us the overall health of cells in a population. And this one I think is really important, and I wouldn't have thought of otherwise, but it can help to optimize cultures. So the media that cultures are grown in, or the parameters regarding experimental conditions. So they can be used for lots of different things, really useful for all different kinds of scientists and experiments, really versatile, and as you'll see, pretty easy. So how are these MTS assays done? So a salt is going to be added to the culture, just straight into the culture, and it kind of turns it yellow, and then any viable cells in the population will start to break down the salt. The salt in this case is tetrazolium, and it's going to break down into a chemical called formazan, and formazan will change the color to kind of like a purplish, and then the purple solutions are what's going to be used to read absorption. So the darker the color, obviously, the greater viability in the population. So we'll kind of talk about the basic steps just here over this visual. So first, obviously here, the cells are just going to be loaded into the wells and prepared. And then cells are going to be incubated. Pretty straightforward. And here we see the purplish blue of the formazan production. And I do have to point out here, this solubilization step is an additional step. So this, I guess, little visual is from an MTT assay, which is almost exactly the same, except that in an MTT assay, the formazan that's produced is insoluble, which is why we have a solubilization step here. But in MTS assays, the formazan that's produced is actually soluble in the cell media, so this step is actually not required for the MTS assays, which is actually another reason you'll hear MTS assays be called one-step assays, because pretty much you load them, you put the chemical in, and then the rest kind of does it for you. And then, so finally, after the formazan is produced, it's going to be put into this spectrophotometric measurement machine. And actually the parameters are slightly different on MTS assays. This one, as I said, is an MTT, and so this range is slightly higher for MTT assays, but nearly the same. So how does the MTS work? So viable cells contain NADH-dependent enzymes. And if you recall, NADH, NADPH has a lot to do with mitochondria and ATP production. So it makes sense that these dependent enzymes are only useful in viable cells. So the viable cells contain this NADH-dependent enzyme, and it's going to break down the salt, and it's going to produce the color change. So as you can see here on the left, this is our MTT, so this is going to be our salt. The the tetrazolium, and then you can see here the NADH loses the hydrogen and formazan is produced, which is how we're going to see that purplish color, which is where we get the readouts. Just some sources. I hope you guys learned some.